Hi, class. Welcome back. We're in the middle of chapter 2. We're actually coming near the end of it. We've discussed some chemical reactions. We've started talking about organic molecules. We've discussed carbohydrates and lipids. And now we're into our next category of proteins. Proteins are the most abundant and the most important organic molecules. They are doing a lot of the stuff that is happening in your cell. So when we talked about enzymes, they were proteins. When we discuss channels in a cell, when we discuss antibodies, when we discuss anything doing really any kind of work in a cell, it's probably going to be a protein. Proteins contain carbon, hydrogen, and oxygen, and they also have nitrogen in them. The monomer of a protein is an amino acid. There are 20 amino acids that will be these monomers or building blocks. And all amino acids have this same structure to them. They have a central carbon atom, and at one side is a hydrogen atom bonded to it. At one side is an amino group, so this NH2 amino group. At one side is a carboxylic acid group, or this COOH, and then an R group. An R group means the rest of it. It's the side chain. That R group is going to be different in the different types of amino acids. So there are 20 different R groups to go with the 20 different amino acids. At some point in your career, if you are going into biology, you'll learn more about R groups later. They could be as simple as a hydrogen. They could be a ring. They could be a carbon. They could be a sulfur. They could be anything else that comes off here. So that is the structure all amino acids are going to share. Amino acids can function in two mirror images of each other, but often we're going to be using this L form. Although honestly, I am never going to ask you this question on a quiz or exam, and you're probably never going to be asked that in real life either. To connect two amino acids, our little monomer here, to connect the monomers, we do dehydration synthesis. So two amino acids. Here you can see the R group here is hydrogen. The R group here is CH3. So there's a little bit about what those R groups are. Anyway, to connect these two, you do a dehydration synthesis. And when they connect, they have just a regular old covalent bond, but it gets a special name called a peptide bond. That's because sometimes we refer to change of amino acids as a peptide. Okay, to break apart two amino acids to their individual pieces is a hydrolysis reaction. Proteins have a lot of jobs in the cell, and depending on what book you're looking, this is probably going to be described differently. But let's go through how the Tortora textbook discusses it. The first possible role is support. So structural proteins, things like actin, that give your cell its shape. Movement or contractile proteins, things like flagella are going to be based a lot in protein. Transport, so these things, letting things go in and out of the cell, whether it's just an empty old channel or a specific little kind of open and close kind of channel. These are transport, those are proteins. Regulating pH, we already discussed how the amino group and how the carboxylic acid group can be involved in regulating pH. Enzymes are all proteins. We discussed that a little bit earlier in this chapter. Hormones, your body's ability to coordinate and control for any multicellular organism. These are proteins. And then antibodies, the way our bodies defend ourselves against infection. These are also proteins. Proteins can be conjugated. That means you can stick other kinds of organic molecules onto it. If you have a protein with a sugar attached, glyco means sugar, you have a glycoprotein. If you have a protein with some nucleic acid, it'll be a nucleoprotein. A protein with some lipid will be a lipoprotein. One very important feature of proteins is their shape. The shape is going to completely define what it fits together against, how it can interact with other molecules. So understanding a protein's shape is very important to be able to understand what it's doing. So you need to be able to describe to me each level of protein structure and what each level is doing. So primary structure, let's actually look at a picture. Primary structure is just looking at this series of amino acids. Leucine, glycine, aspartic acid, whatever all the amino acids are. Just name off the amino acids. That's primary structure, just beads on a string. Secondary structure is when that backbone, when the amino and carboxylic acid groups start to interact with each other. And they form two main shapes. One is an alpha helix, this kind of twirly, twisting shape. And one is a pleated sheet. Sometimes it's called a beta pleated sheet or a beta sheet or a beta folded sheet. But this folding kind of action. This is all going on because of interactions of that amino acid backbone. Then these twists and sheets start to fold up into some three-dimensional. So I think of tertiary means three-dimensional structure we have going on here. This is going to be things like hydrophobic molecules, hydrophobic R groups are going to turn into the middle of the protein away from water. Hydrophilic R groups are going to turn out towards water. Different R groups are going to interact with each other to form maybe polar interactions or even covalent interactions. If you have a couple of sulfurs, they'll make a sulfur bond with each other. You might have just different R group interactions are what's really forming your tertiary structure here. All proteins have a primary, secondary, and tertiary structure. Then some, but not all proteins also have a quaternary structure. So quaternary means this one piece by itself can't work. It has to have multiple pieces together. So here, if we're looking at hemoglobin, it has two of these light pieces, two of these dark pieces. That's its quaternary structure, how different peptide groups fit together. If you're looking at keratin, it has long strands twisted together. Gives it nice strength to give it some support and strength. So that's its quaternary structure, having different peptide groups folding together into one final functional unit. Another thing about proteins is that all these interactions holding them together, they're hydrophobic or hydrophilic or hydrogen bonds or all those bonds that hold it in its shape, can break. If it's too hot, if it's too acidic or basic, those things can break. And if those hydrogen bonds break, you have something called denaturation. In denaturation, never mind. In denaturation, the protein just loses its shape. Instead of being this nice folded up shape, it's just a long old strand again. It's not going to be functional. It won't have an active group. It won't be able to bind to what it needs to bind to or do what it needs to do. So when a protein is denatured, it's non-functional. When you lose your structure, you lose your function. So protein structure gives it its function. You lose a structure, you lose the function. An unfolded protein is not functional. Proteins can be fibrous, which means shaped like a fiber, like this keratin here. Or they can be globular, which means, a glob is something, or a globe, a more soluble sphere. The function that a protein has is going to be based on its shape. So here, this is something that needs to go through your blood to deliver oxygen. So this nice globular shape allows it to just kind of flow through blood. This is something that needs to be a little bit more structurally sound. So that nice, long, tied together makes it a strong rope to support your cell. Okay, and then denaturation again, super important topic. Proteins can be denatured. That means that proteins are in some hostile environment. It's too hot. There's too much acid around. It's too much of something going on, breaking its bonds. When those hydrogen bonds are broken, they lose their shape. And when it loses its shape, it loses its function. So now you can tell me, what are the two functional groups in all amino acids? What are the major functions of proteins? And what are the 4 levels of protein folding? Okay, I'll see you back for our last discussion in Chapter 2, where we are going to discuss nucleic acids. See you later. Bye.